So determination happens before differentiation. Okay, it takes some time for a cell to move from being determined to a fully differentiated cell. And when that happens, we will be able to see actually the cell is becoming differentiated because its shape and structure will change. Okay. Now, cell differentiation will happen when the cells start producing what we call tissue-specific proteins. Okay. These tissue-specific proteins are responsible for giving the cell its unique phenotypes in terms of its structure and function. So a cell becomes determined at the level of gene expression, which genes are expressed. It will become determined because there are certain activators available inside the cell, and these activators will start expressing specific genes. And when these specific genes are expressed, they will give tissue-specific proteins, and these tissue-specific proteins will be responsible for the specific structure and function carried out by the cell. And when that happens, we would say that the cell is now differentiated. Okay. We can understand the difference between determination and differentiation at the molecular level, at the level of the different types of activators available for the cell at the stage of determination, and the different types of genes that are expressed when the cell becomes differentiated. Cells become differentiated because they will express certain sets of genes. We have a sequential, or we have a program of gene expression that involves sequential expression of genes, which means that some genes will be expressed in order for the cell to become determined, and then later on other genes will be expressed in order for the cell to become fully differentiated. And typically, cell differentiation, the differential gene expression, is basically induced at the level of transcription. So transcription is the major mechanisms by which a cell will have a specific set of genes expressed in order for the cell to become fully differentiated. And we could understand the role of differential gene expression and the regulation of gene expression at the level of transcription and its relationship with determination and differentiation by looking at an example. So these two different cells, they are fully differentiated cells, and they are fully differentiated cells because they express different genes. This liver cell expresses the albumin gene, this lens cell expresses the crystalline gene, and there will be other genes that are differentially expressed between these two cells. So there are maybe hundreds of genes that are expressed in liver cell but not in a lens cell, and vice versa. So you will have hundreds of genes expressed in a lens cell but not in a liver cell. And these differences in gene expression are causing this cell to differentiate into a lens cell, and this cell to differentiate into a liver cell. So now we have a specialized liver cell with this structure that is, and it's specialized in making albumin. And now we have a differentiated lens cell looking like this, and it is specialized in making crystalline. So here we have a specific gene expression program, and here we have a different gene expression program. Now, in terms of how this gene expression program happens, it happens sequentially. So if we look at an example, for example, how muscle cells, they become determined and then differentiated. Muscle cells, for example, they originate from an embryonic precursor cell. So an embryonic precursor cell will be expressing some genes, but one of the genes that is important for the development of muscle cells is a gene encoding a transcription factor called myelin. And then we have other muscle-specific genes. So remember, when a cell becomes differentiated, it will have tissue-specific proteins, and these tissue-specific proteins are encoded by tissue-specific genes. So, for example, we have something called muscle-specific genes. These muscle-specific genes will encode muscle-specific proteins that are only, and these proteins are only found in muscle cells. In the same token, you have, for example, liver-specific genes encoding, for example, liver-specific proteins. And these proteins will be only found, for example, in liver cell. And these proteins are responsible for giving the liver cell its functional and structural characteristics. Okay. Now, in the embryonic precursor cell, a master regulatory gene or a transcription factor important for the differentiation of muscle cells, its transcription is off, and the transcription of genes that are important for muscle cells are also off. But when an embryonic precursor cell transcribe the master regulatory gene MyoD, MyoD encodes a transcription factor called MyoD protein. If this gene is expressed in any, of course, we're looking here at one cell. This is not the zygote. We're talking about one cell out maybe of dozens of cells that are found in the embryo. But this particular, so there are other cells, let's say like this, within the embryo, okay? But this particular cell, for some reason, because maybe of a specific cytoplasmic determinant, or maybe because of induction from another cell, it will, at some point, start transcribing this gene. Now, this gene, if it's transcribed, it will produce a transcription factor. And if this transcription factor is produced inside the cell, now the cell is transformed from an embryonic precursor cell into what we call a myoblast. So a myoblast, it's a still undifferentiated cell. As you can see, its shape looks like the shape of, or its structure and shape looks like an embryonic precursor cell. So if you look at this cell under the microscope and this cell under the microscope, both are identical. But at the molecular level, but inside, this cell does not express MyoD, and there's no MyoD. But in this cell, we have transcription and expression of MyoD, and we have MyoD gene, and we have the MyoD protein. If the MyoD protein is produced, now the cell becomes a myoblast, not an embryonic precursor cell, and now the cell is determined. What do I mean by determined? This cell will eventually become a muscle cell. Why? Because this transcription factor, which we could call activator, will eventually binds to the control elements of another transcription factor. And this transcription factor, when it's produced because of the MyoD, so the MyoD activates the expression or the transcription of this gene, and this gene, once it's transcribed, it will produce another transcription factor, and this transcription factor will activate the expression of muscle-specific proteins like myosin and other muscle proteins, and these proteins will be responsible for giving the muscle cells its unique structural and functional characteristics. And now, once these muscle-specific proteins are produced, the cell is fully differentiated and it is called a muscle cell. And now we can see that it looks like a muscle cell. So as you can see here, we have a sequential program of gene expression. Sequential means تدريجي. This gene is first expressed, which in turn will express this gene. Then this gene product will express other proteins that are important for the differentiation of the muscle cell. Okay? So when these proteins are produced, and this protein is produced inside the cell will transform from a myoblast into a fully differentiated muscle cell. Okay? MyoD is called the master regulatory gene because it will drive the expression of transcription factors that eventually will commit the cell to becoming skeletal muscle. So this MyoD is, let's say, in this example, it does not just only drive the expression of this transcription factor, it will drive the expression, for example, of several activators, and these activators, okay, will not only just one, just for simplicity here it's shown one, and these different transcription factors or activators will start driving the expression of the muscle specific genes to produce the muscle specific proteins. Okay. This MyoD is called the master regulatory gene because its presence inside the cell will cause the cell to become eventually a fully differentiated muscle cell. You have to understand that this embryonic precursor cell could potentially become, let's say, a fat cell, a muscle cell, or maybe a cartilage cell. So cartilage cells, muscle cells, fat cells, they all originate from the same embryonic precursor cell. Okay. But what determines if this cell will become a muscle cell, cartilage, or fat cell is the type of master regulatory gene that will be expressed or transcribed in this cell. So in our case, if MyoD protein is produced, this cell will become a muscle cell. If, for example, another protein or another master regulatory gene, I don't know what is the master regulatory gene for cartilage, but maybe if protein called X is produced inside this cell, a cell will be committed to become a cartilage cell. Maybe another protein Y, a master regulatory protein Y, is produced inside the cell, it will later on become a fat cell. Okay. And of course, this MyoD, if it's produced, we know the rest of the story. And you have to understand that scientists call this master regulatory gene regulator because it turns out that certain types of cells you can basically switch, let's say, or what you can basically change the fully differentiated cells, some kinds of fully differentiated non-muscle cells into muscle cells if these cells start producing MyoD. So scientists made an experiment where they took fat cells which are fully differentiated, okay, and these cells, they do not normally produce MyoD, so they are MyoD minus. They do not express the MyoD gene, but they could be converted into muscle cells if they are forced to start expressing MyoD. So the presence of MyoD inside fat cells will convert fat cells, which are fully differentiated, into muscle cells. Same thing applies to liver cells. But this does not work for all types of cells. Some types of cells, even if they start expressing MyoD or they are forced to express MyoD, they are not converted into muscle cells, which indicates that MyoD by itself, okay, might not be, might not work for all cells. Some cells, we can only convert them into muscle cells in the presence of MyoD and maybe other types of activators or other types of master regulatory genes. Okay. So far we have discussed how the differential gene expression will result in differentiated cells and maybe tissues. But in order for a cell or a tissue to function, these tissues and cells that are differentiated should be placed in their proper location, which means that before a tissue or a specific type of cell can carry out its unique function, it must be also located in the right place. So what we mean by that is that in the 3D structure of the organism, every cell must be in its correct location, in its correct place, and it must be, let's say, differentiated, specialized in structure and function. So cells are differentiating during development, but also they are being located with respect to the 3D structure of the embryo. They are being located in the right place. So a muscle tissue or a muscle cell, if we go back to this figure, it shouldn't be located here with the eye cells. Okay, it might be located here, where this part will become, for example, a limb, and a leg will be produced, or legs will be produced from this part. Okay, an eye cell, as this zygote start to divide into cells, and cells becoming more specialized and differentiate, some of the cells differentiate into eye cells will be actually placed in their correct location with respect of the body plan, and they will be located here. So now they are specialized in function, structure, and they are located in the correct place with respect to the organism body plan. So next we're going to learn how actually the organism body plan, okay, is set up during embryogenesis. So the body plan, this basically will determine which part of the embryo will become the right part, right, left, let's say. Which part will become the left, which part will form the head, which part will form the tail of the embryo, which one will form the back, which part will form the front. Okay. And you will be surprised that this body plan, okay, will be established as the cells are differentiating, as the cells are continuously dividing at a very early stage of development. Okay, so we have the body plans. Let's say we have, we could say we have right, left, head, tail, and then we have let's say back and front. Okay, or we could say this is anterior and this is posterior, and the back is basically dorsal. This is the scientific name, and the front is ventral or belly. Okay, so the cytoplasmic determinants and induction signals, not only they contribute to the differential gene expression and cell differentiation, but also they contribute to the development of a spatial organization of tissues and organs. Spatial means 3D organization, where tissues and organs will be located within the body plan. And this process is called pattern formation. Okay, and you have to understand, pattern formation begins with the establishment of the major axes of the body. These axes, محاور. Axis means محاور. These axes will determine which side will be the head or the anterior, which side will be the tail or the posterior, the right, left, and back and belly, or dorsal and ventral. And we have molecules that are important for establishing these major axes. And these molecules are responsible for controlling pattern formation. These molecules are called positional information. Okay? Positional means مكاني or مكان from مكان. And these positional information, again, they are provided by cytoplasmic determinants found in the cytoplasm of the egg, and by inductive signals, molecules produced by some embryonic cells that will induce basically cell differentiation and morphogenesis in other types of the embryos, in other types of cells of the embryo. Okay. These molecular or molecules, the positional information molecules, they will tell cells their location relative to these body axes. Okay. And relative location compared to other cells or neighboring cells. And then they will determine these molecular positional information, basically how a cell progeny or the cell daughter cells produced from that cell will respond eventually to future molecular signals. So scientists began, even before they discovered that genes are made of DNA, scientists in the first half of the 20th century, they start studying how organisms undergo development, okay, by looking at just how different embryonic stages they look like. So even before scientists could study the DNA or study the gene expression or which genes are expressed, which genes are not, they focused on studying the shapes or the structures of different stages of embryo, how the zygote looked like, how different stages of the embryo will look like. Okay, but this type of research at that time couldn't explain why a cell will become a muscle cell in the future, or why a liver cell, or why a particular cell, let's say, why this particular cell, for example, will become an eye cell in the future, or why this particular cell will become a mouth cell. Of course, we know now that these cells, they are expressing different genes. But in the 19th century, they couldn't, do experiments to understand at the molecular level why this cell become an eye cell and why this cell become a mouth cell. And not until that the scientists discovered that the genetic material or genes are encoding proteins and genes are made of DNA. So later on, scientists start making experiments at the DNA level to try to understand how to link, the different, let's say, different structures of cells within the, at the different embryonic stages, how the DNA or how gene expression, differential gene expression will cause cell differentiation and morphogenesis and cause basically the development of embryo into an adult organism. Pattern formation, the development of the bloody axes, have been extensively studied first in the fruit fly Drosophila melanogaster. Okay. And then scientists began to study the genetics of this fly and the genetic basis of embryonic development in this fly. And later on, scientists, they combine both the phenotypic, genetic, and biochemical approaches in order to fully understand how a Drosophila melanogaster zygote develops into an adult fly, for example. Okay. So in the next lecture, inshallah, we are going to be looking at how melanogaster zygote will develop into an adult fly, and how the differential gene expression will drive this developmental program of the melanogaster fructa. And what scientists learned about, when scientists learned about the details of the genetic details or biochemical anatomical details of development in fruit fly, they found that the same principles could be applied to other species like humans, for example. Okay. So far we've learned about the role of cytoplasmic determinants. Determinants and inductive signals. Both, they are important for the differential gene expression in the cells of the embryo or developing embryo. And this differential gene expression will allow the cells to undergo differentiation and morphogenesis. Cell differentiation, cell determination, and morphogenesis. Also, these cytoplasmic determinants mainly are important for what we call pattern formation. So these cytoplasmic determinants and inductive signals both are important for pattern formation, okay? And both they are called positional information. So in pattern formation, what will happen is that every cell will know its place within the organism. So we have establishment of the major axes of an animal, okay? The head, the tail, the right, the left, and the back and the belly, okay? So in pattern formation, we have basically the major body axes are formed because of these cytoplasmic determinants and inductive signals that are basically start acting or influencing the development of the embryo even at a very early stage during embryogenesis. So we have in pattern formation, we have basically the three major axes, body axes, are even, let's say, they start to appear or organized even at early stages of development because of these cytoplasmic determinants and inductive signals. So today, inshallah, we're going to learn about this pattern formation and the role of cytoplasmic determinants in pattern formation by looking at an example organism. The example organism we're going to be discussing is the Drosophila melanogaster. Drosophila is the fruit fly. It was its development and the role of genes, gene expression, cytoplasmic determinants in the development of Drosophila flies have been studied extensively by scientists. That's why it's a good model. It is considered a good model to study, development or embryogenesis, and to study the genetics, the biochemistry of embryogenesis or development. So today, inshallah, we're gonna look at the life cycle of Drosophila. Then we will start discussing how a zygote of Drosophila fly develops into an adult, and what is the role of the cytoplasmic determinants and even the genes of the embryo during development. So this figure shows the development of a Drosophila starting from an egg into a larva. The cytoplasmic determinants in Drosophila in the egg, in the unfertilized egg of the Drosophila, as I told you in the previous lecture, they establish the axes or determine the axes even before fertilization with the sperm. Now, after fertilization, as you can see, after fertilization, so before fertilization, we have a developing egg, and the developing egg consists of a single cell with a nucleus surrounded by follicle cells and a nerve cell. So these nerve cells, they provide the egg, the future embryo, with nutrients, with mRNA, with certain proteins. Now, after fertilization, so this is the mature egg, but it's still unfertilized. This is a still developing egg. After fertilization, when the females start laying eggs and the eggs are fertilized, they will start they will start their own developmental program, and the fertilized egg will start undergoing several rounds of cell division, mitosis. At the same time, we will have, of course, cell differentiation and morphogenesis. So here we're talking about embryonic development. This means we have cell division, cell differentiation, and morphogenesis. Eventually, we will have an embryo, okay, and this embryo is considered segmented embryo, segmented muqatta, which means it consists of segments, as you can see here, different segments, and they are color coded to indicate that these certain segments will eventually form the head. Other segments that are colored brown, they will form the thorax, from which the fly will have the wings and the legs, and then these orange segments, they will form the future abdomen. And then we will have a segmented larva, dude. Okay, and eventually this larva will form a pupa, and then metamorphs into an adult fly. So this is basically a picture of the adult Drosophila fly. We have three major parts: the head, the thorax, the abdomen. And this chart on the left, or this cartoon on the left, shows the body axis. We have the right, left. Dorsal means here back, ventral, the belly, anterior, the head, let's say, or the front. The posterior is the end of the, let's say, tail. Okay, and of course, right and left. And Drosophila, they are considered bilateral organisms. So they are bilateral, symmetrical organisms. So bilateral means we have two sides, right and left, and then these two sides, right and left, they are identical. Okay, so humans are considered also bilateral organisms. All right, so let's talk about how scientists analyzed the early development of Drosophila eggs into larvae and into adults, and how scientists used relied on genetic analysis, okay, to determine which genes are important for development in Drosophila. So in 1940s, scientists started using, let's say, genetic approaches, study the genetics of Drosophila embryogenesis or Drosophila embryonic development. And the first or the pioneer scientist who started doing that was Edward Lewis. So Edward Lewis, what he did, he wanted to know, okay, he wanted to discover the genes that are important, let's say, or genetic loci, okay, that are important for the development, embryonic development in Drosophila. And his approach involved introducing mutations into the adult fly, and then he allowed fly to to mate, and then he looked for any bizarre, let's say, mutants with developmental defects. So the idea here is that if there's a fly, okay, and this fly, let's say, let's see, okay, these flies will be mutagenized. So what he did, Edward Lewis, he introduced mutations into adult flies. So this is adult fly. And he allowed these flies to mate. Now these flies will produce what? When they are mated, again, they are mutant flies, so they have mutations in some of their genes. Okay, he didn't know where the mutations, which genes will be affected by he didn't know which genes might be affected by these mutations. And after mating these mutant flies, they will produce progeny. Okay, and these progenies, let's say this is a larva, these progenies might have what we call developmental defects. Developmental defects, or let's say abnormalities, means that these larvae or these progeny, they have some problems and they cannot develop normally because they inherited genes that are mutant from their parents. And these genes, if they are important for development, and if they are mutant genes or mutated genes, they might affect the process of development, and the larva might not develop properly. Okay. So what he did, or they might develop into adult larvae, but they will have some let's say. So he was looking for mutant phenotypes. Which means after mating and after the development of the progeny into adults, okay, these, let's say, these mutant, or the progeny that produced from crossing or mating mutant flies, they might have some, mutant phenotypes where they might be missing some parts, or they might have some parts in their incorrect places. So we have a picture here, for example, of a wild type head of a Drosophila fly. So here, this is a Drosophila fly head. So this picture, we have Drosophila fly head. And as you can see here, we have the eyes. Okay, maybe this is the mouth, and I'm not expert in the anatomy of Drosophila fly. But here we have what? The fly antenna. Okay. Now, when Edward Lewis did his experiment by mutagenizing flies and mating them, and then he analyzed carefully the resultant progeny, he was able to find some bizarre mutants or flies with a mutant phenotypes. For example, he he obtained some mutants where instead of having an antenna here in the head, they had legs. Okay. So what he did after that, so this is a mutant phenotype. The next step was to locate the mutation that caused this phenotype. So his approach was simple: mutagenize the flies, cause them, obtain mutant phenotypes, then determine which genes or which genetic loci were responsible for these mutant phenotypes. By using this approach, Lewis and later on other scientists, they all use similar approaches to discover the genes that are important for the development in the late embryo, larva, and adult stages. And they call these genes homeotic genes. So now we know that the genes that are important for the development in fruit fly in Drosophila, they are called homeotic genes. Typically, these genes are they control pattern formation. They control basically they are responsible for establishing the major body axis, and they are important for telling every cell, every structure, or every tissue where it should be, where its correct place in the final, structure of the adult organism. Now we know that there are around 1,200 genes that are important for pattern formation in Drosophila. Okay. And of these 1,200 genes, we have 120 genes. They are essential for normal. Well, it's written here. So 120 genes essential for normal segmentation. So these genes are important for the correct formation of all segments in the larvae. Okay, and later on, of course, in the adult. Now, again, the first scientist who used the genetic approach to discover the genes important or genetic loci important for embryogenesis or embryonic development of Drosophila was Edward Lewis. Later on, Pollard and Weisstrass, also they continued the work of Lewis, and they used a similar approach to determine basically all the genes that are important for, let's say, pattern formation and important for segmentation, correct segmentation or normal segmentation in the late embryo, larva, and the adult stages. Now, you have to understand it wasn't an easy thing to do because of many, so these three scientists, they faced many challenges when they were trying to determine which genes are important for pattern formation and segmentation, or what genes are important for normal embryonic development. Because you have to understand that there are a lot of genes in Drosophila. So Drosophila contains around 1,400,000 genes. Okay, and this is a lot of number of genes to study and to basically to determine which of these 400,000 genes are important for embryonic development. Okay, so here, which genes are important for, let's say, for pattern formation or segmentation, or in general for development. Again, their approach was to mutagenize these genes, some of the genes, and look for mutant phenotypes in the progeny, at the larva, at each stage of development. But you have to understand that many of the mutations they introduced into the flies, they were embryonic lethal. Okay. Embryonic lethal means cotton. Many of the mutations they introduced into the parent flies did not result in a viable progeny, which means many mutations they actually were lethal. Embryonic lethal means that the embryo will die, will not basically proceed to develop into a larva, then into an adult. So, because certain mutations in certain genes, okay, if mutations hits like an essential gene, it might destroy completely the function of that gene, and if that gene is essential for development, the embryo will not be able to develop. Okay, so some mutations. So I'm listing the reasons or some of the challenges faced by these scientists when they started studying, segment formation and the relationship between genes and segment formation. So some mutations are embryonic lethal. And they cause the death of the embryo. That's the phenotype. That's the phenotype of these mutations of the embryo. And of course, if the embryo dies, it will not show its mutant phenotypes, and they cannot study it. So this is one of the challenges these scientists encounter, and they were able to to go around or find a solution for this problem by inducing these mutations that are embryonic lethal, but they were looking for recessive mutations. Okay, so these recessive mutations will be found in heterozygous flies. Okay, and heterozygous flies, they will have 2 copies of each gene, one mutant copy or allele, let's say, of any gene, and the other is a normal copy. But if the mutation is recessive, the presence of one normal copy of the gene is enough, and it will not cause death. So the idea here, instead of inducing embryonic lethal mutations in both copies of the gene, they will induce a mutation, or they will look for heterozygous flies or mutant flies that have only mutation in one of the alleles of the gene, and that mutation should be recessive. It shouldn't be dominant. So a dominant embryonic lethal mutation will cause death. A recessive embryonic lethal mutation will not cause death in heterozygous flies. Okay, a dominant embryonic lethal mutation will cause death in heterozygous flies, but a recessive one will not cause death. So they looked for the analyzed flies, heterozygous fly, carrying these embryonic lethal mutations that are recessive, and looked for the phenotype, and then determined where the mutation it was located at the genome, and which gene was actually affected by the mutation to cause the mutant phenotype, the non-lethal mutant phenotype in heterozygous flies. Sorry, I did not check the chat. So we have question from Isra 10 minutes ago. زي الطفرة بتكون من فقدان كروموسوم X لإكتراز كروموسومي Y. So they found the mutant gene from the larvae phenotypes. Yes, يا تالا. طيب دكتور كيف لاحظ هاي الطفرات وهي recessive? So they noticed that some flies, they had a mutant phenotype, and if you have, for example, let's say if you have a mutation, if we have a gene, and this gene is gene X, and gene X is essential, let's say. Essential means essential أساسي means it is important for life. If this gene is non-functional, the organism will die. So let's say that there's a type of mutation that could affect gene X. But you have to understand, Drosophila flies, they are diploid organisms, which means they will have 2 copies of gene X. Let's say that there are 2 types of alleles of gene X. Allele X, which is normal, and let's say we have allele X star. And here we have a mutant allele, and this mutation is recessive, okay, mutation. So imagine we have 2 heterozygous flies, X, X, well, and then we have X, X. So 2 heterozygous, which means each, so this is, let's say we have male and female. Now, each one of these, this is the genotype of both flies. They are heterozygous for gene X. Each one of them will have a normal allele and a recessive allele. But in terms of phenotype, both are normal. Imagine if these 2 flies are mated or bred, what we will have all possible possibilities. So we could have progeny that will have both normal alleles, progeny that will have 1 normal and 1 mutant. Okay. And then progeny or offspring that have 2 mutant genes, that this is embryonic lethal. Maybe they are embryonic lethal. So they will, but here we will have just let's say mutant phenotype. So this progeny will live, but it will have a mutant phenotype. Maybe it will show a mutant phenotype during development. Okay, but later on it might, develop into, a normal. Well, it's you have to understand that they mutagenize these flies. So these flies, they are normal, but once they are mutagenized, they become, let's say, heterozygous. Okay, flies, and then they will focus on studying the heterozygous, because the heterozygous flies, they might show up a mutant phenotype, and they have this mutation in this gene that is important for development. Okay, but if some offspring or progeny, they acquire both copies, okay, of the mutant fly, they will die and they will not be able to continue with their developmental program. Okay, this is what I meant by studying recessive mutations rather than, in heterozygous flies, rather than studying the mutations in homozygous flies, because some of these mutations might be embryonic lethal. Okay, you have to understand also, these scientists did not only study the genes of the embryo, they have to study the genes of the mother. Why? Because we know that mother genes, the genes or the maternal genes, they are encoding the cytoplasmic determinants in the egg. And we know that these cytoplasmic determinants in the egg, they are important for cell differentiation and for pattern formation. So not only these scientists had to focus on the genes of the let's say the embryo, but also they had to focus on the genes of the mother, because the genes of the mother, they encode the cytoplasmic determinants that are found in the egg, and these cytoplasmic determinants are important for the early stages of the development in the embryo. Also, they are important for pattern formation. Again, these are some of the challenges that they faced when they tried to determine all the genes important for Drosophila development, and eventually they were able to found 120 genes that are essential for normal, let's say, segmentation during embryonic development in Drosophila. And any mutation in these genes, it might have an effect it might cause an abnormal or unusual segmentation, leading to mutant phenotype or bizarre or weird phenotypes. Okay. Now, with the work of these three scientists, scientists were able to draw a whole picture of the let's say genetic basis of Drosophila development, and this is why these scientists were awarded the Nobel Prize in 1995 for their contribution in decoding pattern formation and discovering the genes that are important for pattern formation, normal segmentation during embryonic development in Drosophila. I told you before that cytoplasmic determinants that are found in the egg even before fertilization are important for pattern formation and are important for establishing some of the body axes. Now let's have a look let's look at an example of a cytoplasmic determinant found in the egg, and how it can basically determine or establish pattern formation or one of the axes. So we have what we call maternal effect genes. These genes, they encode the cytoplasmic determinants found in the egg even before fertilization, and these genes, therefore, they are genes of the mother. They are called maternal effect genes, but they are responsible for determining the axes in the progeny or in the offspring or in the body of the embryo. Another name for these maternal effect genes is egg polarity genes. Polarity means قطبية. They are called egg polarity genes because they will determine within the egg the different sides, or let's say they will divide the egg into different sides. So one side will be, for example, the head, the tail, maybe the back, the belly. That's why they are called egg polarity genes. They will determine the different poles of the egg, which side will be the head, which side will be the tail. So eventually they will control orientation. Okay, اتجاهية. Let's look at an example of a cytoplasmic determinant that is encoded by a maternal effect gene or an egg polarity gene. This gene is called one of the genes is called bicode gene, and it is important for establishing the anterior posterior axis. It will determine which side of the egg will become the front, which side of the egg will become the back or the tail. Okay. So this bicode gene is important for establishing the head to tail axis. Now, the bicode gene encodes a protein called bicode. And the bicode, of course, we have a bicode gene which will be transcribed into bicode mRNA and then translated into bicode protein. When scientists looked at the bicode mRNA and the bicode protein, they found that it's more concentrated at one side of the egg compared to the other. So this figure shows, so here we have unfertilized egg, just drawing a line. And this here, this region contains what? Contains the bicode mRNA. And here we're looking at the location of the presence of the bicode protein. Notice it is found at this end. And this end, eventually it will become the anterior end, which will give the head, for example, later on in the adult fly. And here, the other side will become the tail or the posterior end. So before fertilization, this bicoid protein is not present in the egg. There's only bicoid mRNA, which is more concentrated at one end compared to the other. After fertilization, as I told you before, protein translation will begin, and the bicoid mRNA will be translated into bicoid protein, and still the protein will be more concentrated at this side compared to the other. Okay. And this bicoid, which is a product of the mother genes, bicoid, was found to be essential for establishing the anterior posterior axis. It is important for establishing the anterior end of the embryo. Wherever it is located, this part of the embryo will become the anterior end or the head. Okay, and the opposite part will become the posterior or the tail. Now, this bicoid is also called, is an example of a morphogen. So a morphogen is a substance that is found inside the embryo at different concentrations along the length of the embryo. So we could, for example, look at the concentration. So we have a concentration gradient of the bicoid mRNA and the bicoid protein in the embryo. On this side, on the left side, we have a high concentration. So this is bicoid mRNA concentration gradient. Gradient means we have a highly concentrated region of the bicoid mRNA, and as you go from left to right, the concentration decreases significantly until we have no or very, very few mRNA molecules here. Same thing applies here. This is the concentration gradient of the bicoid mRNA. Substances that have a concentration gradients within cells or within embryos, they are called morphogens. Okay? And morphogens were found to establish the embryo's axis. An example of a morphogen is the bicoid. Of course, there's another morphogen that will determine, for example, there might be another morphogen, let me draw it here, that might determine, let's say, the other axis, basically the ventral dorsal axis, or the back and the belly axis. Okay? Axis. So basically, there will be a concentration gradient like this. We have on top a high concentration, as you go down, a low concentration. So this will be the back, this will be the belly. Okay? So these morphogens, they are involved in establishing body axis at early stages of development. Okay, an example of a morphogen is the bicoid protein, and remember, the bicoid protein is also a cytoplasmic determinant. Okay, so the bicoid protein, it's a morphogen. It's a cytoplasmic determinant. So don't be confused. We're talking about the same molecule. Also, it is encoded by maternal effect gene or egg polarity gene. Okay, so all of these are characteristics of the same molecule, the bicoid molecule. Okay, questions so far? خلينا نكمل. So we have a question from Aziza. Sorry, I cannot get what is a morphogen. Is it a gradient of substance? A morphogen is just any substance that will have a concentration gradient, along the length of a cell or an embryo. Okay, so here, if we looked at the bicoid, here we have a concentration gradient. So the concentration of the bicoid protein here is very high, and as you go from left to right, its concentration will begin to decrease until you have nothing at the other end. That's what is meant by concentration. So here, for example, this is so here we have a picture of a wild type larvae, a normal larvae with the head, the different segments, and then a tail. This is a mutant larvae. Okay, so this larvae is mutant, a mutant larvae because of the bicoid gene in the mother was mutant. So this bicoid, this larvae, okay, will develop two tails because there is no bicoid proteins that are found in this larvae. Okay, so this is a mutant larvae whose mother has two mutant alleles. So the mother of this larvae had two mutant alleles of the bicoid genes. So there is no bicoid protein in this larvae. This is why you have two tails, okay, formed at both ends of the embryo. So this bicoid, the discovery that there is a morphogen, a cytoplasmic determinant called bicoid, responsible for establishing one of the body axes, was very important, was groundbreaking, because it identified a specific protein that is required for early steps in the pattern formation. And this specific protein is actually encoded by the mother genes. It allowed also the discovery of the bicoid lab also to scientists to understand the role of the mother and the mother genes in embryo development. And we all know this through the because of the presence of cytoplasmic determinants even before fertilization. Also, the bicoid research demonstrated that there are molecules called morphogens, and their concentration gradient is important for determining the body axis or polarity and position in the embryo. Okay. So before we move on into the next section, which is about cancer, please. So the next topic, or the last topic of this chapter, is about gene regulation, the control of gene regulation and cancer. So in this chapter, we're going to learn how mistakes in gene regulation might transform a normal cell into a cancerous cell. So we learned about the regulation of gene expression in normal situation. Here, we're going to learn how we learned about gene regulation and its role in embryonic development. Here, we're going to learn about the role of gene regulation in cancer and how when there are mistakes happening in gene regulation, it might lead to the development of cancer. So we know that cancer can be caused because of mutations happening in certain types of genes. You learned about cancer in chapter, I think, 12 in Biology 101. So cancer is a type of disease in which cells, they start dividing without control. Okay. So remember when you were learning about the cell cycle control and the cell cycle in general and mitosis, every cell needs to be controlled in terms of when it can divide and when it cannot divide. So there is a control system inside our cells that will ensure that cells only divide when they are needed to divide. If this cell division or cell control system, for some reason, something wrong happens to it, cells might start dividing without control, and they become what we call cancer cell or cancerous cell. You have to understand that cancerous cells inside, the human body, they are actually used to be normal cells. So a normal cell can be transformed, or the process of converting a normal cell into a cancer cell called transformation could happen this could happen because of, mutations. Now, these mutations will have different effects, or it might affect different genes that are important in controlling the cell cycle, for example, inside the cell, or in genes that are important for, the behavior of cells, some characteristic of cells. Now, cancer cells that are found in individuals that have cancer, they do not come from outside their bodies. They are used to be normal cells, but they were transformed into cancer cells because of the accumulation of mutations. Now, how these mutations might transform a normal cell into a cancer cell, we will learn about this, inshallah, in this today and maybe next lecture. But typically, these mutations, they affect genes that are important for regulating cell growth and cell division, okay? Or genes that are important for regulating the cell cycle or cell cycle control. Of course, how these mutations are introduced into normal cells? Well, they might happen spontaneously. We have spontaneous mutation. Spontaneous here means tilqai. The chances that a person will, have a spontaneous mutation or mutation in his or her genomes increases with age. So we know that cancer affects older people because older people, they might have been exposed to, they might have accumulated with time more spontaneous mutations. Also, we have some chemicals, radiation, some physical, chemical, or biological agents. If you remember, we discussed this in chapter 17. Some chemical, physical, and biological agents might also cause mutations. So whatever the cause of the mutations, some of the mutations, if these mutations happens in genes that are important for regulating cell growth and division, they might lead to the transformation of a normal cell into a cancer cell. Now, cancer cells, they start divide without control. Normal cells, they divide only when needed. Divide without control means they continue to divide over and over and over. Okay. And when they start dividing without control, they eventually they will form a tumor tissue, and this tumor tissue will interfere with the normal functions of other tissues or organs, leading to maybe eventually to death. And this tumor tissue might also, spread into different parts of our body. So these cancer cells, they might form tumor tissues, and they could spread. Some of them. Spread means انتشر. Tumor tissue, نسيج سرطان, which is an abnormal tissue. أفنان, she's asking, دكتور طيب ليه الكبار في السن معرضين تراكم طفرات أكثر مو كل خلايا؟ Well, with age, you become more exposed to chemicals, radiations, and viruses, and you have to understand that we have some systems in our cells that will correct these mutations and will prevent them from happening. But with age, the chances that you will get these mutations will increase with time. Okay, and you have to understand that some types of cells that are not supposed to divide, okay, but some mutations might cause them to start dividing. Yes, not all cells will be dividing, but some cells, let's say skin cells, some types of skin cells or some types of liver cells, they reserve the ability, so they have the capacity to divide when needed. For example, when you have a cut in your hand from a knife, let's say, if you cut your hand by mistake, blood will go out from the site of the cut, and what will happen is that the skin cells will start dividing to close the area that was cut, so to stop bleeding. So here we're talking about skin cells that normally they're not dividing unless they are needed to divide. When, for example, when you have a cut or jurh in your hand. But the idea here is that some normal cells, they appear not dividing, but they have the capacity to divide when they are needed to divide. And these cells, if they somehow acquire mutations in genes that are essential for cell growth and division, they have the capacity to divide. And if they accumulate certain number of mutations in genes important for controlling cell division and cell growth, they might start dividing even when they are not needed to divide, and they will start dividing until they form a tumor tissue, an abnormal tissue surrounded by normal tissues. Also, they might start spreading to other locations in the body and start forming other types of tumor tissues. Okay. Now, we have two major types of genes that are important for cell, let's say, cell growth and division, cell division. We have oncogenes, oncogenes, and we have tumor suppressor genes. So this arrow indicates that proto-oncogenes, their function normally is to promote cell growth. So here we have these genes, their products, they promote cell growth and division. But also we have tumor suppressor genes, their products, they actually inhibit cell division. So promote means يشجع. Here inhibit يثبط. So as you can see, cell growth and division is controlled by genes that will allow cell growth and division to happen, and by genes that will basically stop the cell from growing and dividing.